Okay. Well, thank you very much for that introduction, and also thank you very much for the invitation. And so I'm going to talk about mathematical modeling to understand collective cell motion. And well, we all know that collective movement happens everywhere. So in ecology, we see with bird flocking, fish schooling, whales, locusts, ants. And in biology, we have them across all three stages, key stages, in normal situations, developmental biology, in repair, and also in disease. And the big question everybody wants to know is, how does this all happen? And so what I'm going to do is I'm going to sort of review two areas that we've been working in over the last few years. Cancer, which is going to be more talking about the sort of modeling and analytical challenges, and then neural crest, which will be more integrated with the biology. Okay, so I'm going to start off with a paper that's actually almost 30 years old now by a collaborator, Bob Gatenby, who's a physicist who became a radiologist, and now he's head of radiology at the Moffitt Cancer Center in Tampa. And his work here with Ed Galinsky. So Bob was one of the first people to start to think of cancer as an ecological disease, in the sense that think of cancer cells as invaders, and the host are the cells, and our body is sort of the earth, if you like to think of it. So if you think of it that way, then you've got already 100 years of research that's been done on how do certain organisms invade other organisms in ecology. And so one of the questions that he was asking was that, well, what you would think is that if you have an invader, then the invader should be stronger than the host, so the invader can fight and win the battle. But it's well known back from the 1930s, the Warburg effect, which is that, well, we all know that when there's not enough oxygen, then cells revert to anaerobic metabolism, which is an order of magnitude less efficient at generating energy. And that's why we get cramp, stuff like that. But the question then is that tumor cells do undergo glycolytic metabolism when there's enough oxygen. So the question he was asking is that why do the tumor cells actively resort to an energy source that is an order of magnitude less efficient in producing energy than their competitors? Doesn't make sense. But one of the byproducts of the glycolytic pathway is lactic acid, and lactic acid is more toxic to normal cells than it is to tumor cells. So in fact, they're not using glycolysis really firstly to generate energy, they're using glycolysis to generate a weapon to kill the host species. So that was their idea. And so what they did was they wrote down, so N1 are normal cells, N2 are tumor cells, L is lactic acid, and so they wrote down the standard competition Lotka-Volterra model, where if there was no N2 and no lactic acid, you'd get homeostasis N1 equals K1. But then you have competition, alpha 1, 2 is positive, you have competition from N2. N2, the tumor cells, same thing, competition from the normal cells, but they're not, he has them in their model, they are immune to the lactic acid, So lactic acid doesn't affect them. And also they move. And this is a nonlinear diffusion term here. The idea being if N1 is at the homeostasis value of K1, then it forms a barrier and the cells can't move. But if some of the N1 get killed by the lactic acid, N1 decreases and the cells, the diffusion becomes non-zero. Okay, so mathematically, this is a degenerate cross-diffusion term. And so as a mathematician, your eyes light up because there's going to be a lot of fun and a lot of highly frustrating days spent trying to analyze this mathematically. And then the lactic acid is produced by the tumor cells. It's degraded by it's sort of general degradation, and it diffuses. Okay, so we don't really need a model, nor do we need to simulate this to figure out what's going to happen. Tumor cells are going to come in. They're going to produce lactic acid. Lactic acid is going to kill the normals. That's going to create space. The tumor cells are going to move, create more lactic acid. Lactic acid diffuses, kills the normals, and you move in. Okay, so why waste our time simulating this? Well, when they did simulate it, they found something that they weren't expecting. This is the verbal model I just gave you. Tumor, lactic acid, kills normal, move in. This just means it moves in. But they found in some regions in parameter space, you got this acellular gap. And you got this sharp front because of the degeneracy of the diffusion equation. So that's something they hadn't expected. So then Bob went back and looked at some head and neck cancers that he had, human head and neck cancers, and what he found, in 1/3 of them, you have this gap, acellular gap. Then they did some experiments on tumor spheroids stuck in a mouse, and then you take cross sections, you look at the pH, and what you find is the pH has the gradient that's qualitatively exactly what their model predicted. Okay?
Acellular, there's a void which is filled with some.
Yeah, that's just filled with matrix. Yeah.
Okay.
So just be matrix here, but there's no cells. So the cells are here and the cells are there, and then there's just the matrix in between. Yeah. So obvious next thing to do, and this is what Bob did with Ian Roby, stick some cancer cells into a mouse that metastasized to the lung, treat some of the mice with bicarbonate, treat the other ones, don't treat them, look at the metastases to the lung, and what you see, there's a significant decrease. Okay, so we did some work with them where we then did a sort of allometric scaling up from mouse to human, and what we found was that the bicarbonate wasn't having the level of impact that it was having here. But then we said, that's no problem at all. We went back to Bob and said, look, Bob, this doesn't work if you just do the upscaling, but that's fine. If you just give this amount of bicarbonate, you will cure the patient. And that's when us idiot theoreticians found out that bicarbonate is toxic. And so the amount of bicarbonate that we were proposing from the model to give would kill the patient. But it would cure them of cancer, right? So Bob's continuing this work because there's other things about, how could you give the bicarbonate in a way and look at the metabolism, et cetera, et cetera. One of the funny things we found in this was, of course, you got a parameter space and you can vary the parameters and everything. And we found one region in parameter space where you could lower the level of bicarbonate and get a good outcome. And then Bob was able to tell us that those parameters that we changed actually corresponded to people having a certain disease. So you could have a situation where an ill person would respond better to the chemo, to the application of this treatment than a healthy person. But then they've gone on and done a lot more work on this and look at metabolism, et cetera, et cetera. What we've done is go and look a bit more at the mathematical aspects of these. Okay? Now, here we showed this nice smooth front. This is in 1 dimension. Extrapolate to 2 dimensions, and you get a nice smooth wave going like this. Most cancers don't have nice smooth wave unless you grow them as spheroids in a dish or in a tube. And I've heard people argue that because tumors have these finger-like protrusions, that means there must be heterogeneity. Now, as a mathematician, that annoys us because implicit assumption is that the 2-dimensional wave is stable. And we all know of examples where you can destabilize a 2-dimensional wave without having heterogeneity, spatial heterogeneity. And so what we did recently with Paul Carter, Arjen Dolman, Peter van Heister, and 3 very talented undergraduate students at the time. So what had happened was Paul, Arjen, and Peter had actually been analyzing this same problem in an ecological setting where they were developing models that showed as vegetation patches grow, you get these fingering-like instabilities. And so I was visiting UC Irvine, I think it was 3 years ago, and we having a chat with them. And then they looked at Bob's model, and then they said, well, but actually, if there was a bistability in the system, then we think we could transfer the analysis that we had for the ecological system onto this model. So is there bistability? Because there isn't bistability in Bob's model. And in fact, I'm sorry for changing the notation here. This is N1, this is N2, and this is L. What Bob actually ended up solving was a 1 minus U and 1 minus V, so they didn't even put the competition in, and that didn't really affect the behavior anyway. But the thing is that there is a controversy, but there's some evidence that there may be an Allee effect going on in tumor cells where there has to be a sufficient number, a sort of quorum of cells before they can grow. So here A lies between 0 and 1. And so if you look at this system here, it's got a stable state at 0, a stable state at 1, an unstable state at A. And if you perturb to be larger than A, then you grow to 1. And that's called Lee effect. So what we did here was take their model, put in the fact that there will be some death due to lactic acid from the normal cell, the tumor cells, do a scaling because the lactic acid is going to diffuse much more faster than the tumor cells are going to move. Here, we removed the singularity so that if U equals 1, this is no longer singular, and there's 2 reasons for doing that. 1 is for mathematical convenience, and 2 is for the biological observation that tumor cells especially the cells at the front, upregulate aquaporins that essentially lead to very stiff filopodia that sort of punch their way through the environment they're moving. So this is an argument that's saying, no, really, even if you do have confluent level of normal cells, the tumor cells can still punch their way and move through. And then we analyze this. I'm not going into all the detail. There's slow and fast variables, et cetera, et cetera. You can do all that, get the different manifolds, et cetera, et cetera. And what you find is, and now these are the tumor cells, just plotted themselves. These are plotted on a co-moving frame, so that really the way you've got to interpret this is that this is really invading in this direction, but the frame is moving, and that's why it looks like these aren't moving, but they are actually invading. And you can see here, we came up with two types of behaviors. One is this cusp instability, and one is this fingering instability. And this is all arising without any heterogeneity in the system. And you see this cusp instability is sort of a little bit like the cusp that you're seeing around here. And we find this happens for the weak Allee effect. That gives rise to fast traveling waves. Strong Allee effect gives rise to slower traveling waves. Now, yeah? No, so this here is just purely qualitative. So it's purely finding parameter spaces in which the only sort of biological thing we've done here is say that this is faster than this. Otherwise, this is all finding, I mean, what you do find is you find quite a large area of parameter space where this happens, and a large area of parameter space where this happens. So it's not, but you're absolutely right. The next step would be to get the data on this. And this is one of the areas which you was talking about earlier on, that it's a real challenge in cancer, is to actually get spatiotemporal data that allows you to fit your model. And there's two reasons for that. One reason is there's no such thing as cancer. There's 200 different types of cancer. And secondly, they're so variable from patient to patient. So to really parameterize one of these, you would have to parameterize it per patient. And that's why what we've been doing here is to try and see, are there parameter spaces in which we see certain types of behavior? But it's a real issue in modeling if you want to really take this sort of stuff to the clinic.
So weak refers to the parameter A?
Yeah, so if A is small, yeah. So it's understandable that if A is small, you'll get fast traveling waves, because that means the tumor can grow quickly, and you get slow traveling waves here. That's understandable. Strong Allee effect. One question that the referee asked was, why do you get these smoother fronts and these fingering? How can you explain that intuitively? And that we really struggled with. We can't explain this intuitively. We just had to say, sorry, this is just what the mathematics shows us. We can't explain it. And the editor accepted that, which was nice. So this paper should appear soon in Journal of Mathematical Biology. So now we ask the question about analyzing the traveling wave behavior of this system. So what we're going to do here is we're going to, and this is work with the graduate student Chloe Colson and colleagues Faustino Sanchez-Godinho, Helen Byrne, Tommaso Lorenzi. So what we did here was we said, now the other thing, in the Gatenby model, he looked at the cells as being a barrier. but there's also matrix. That's also a barrier. So what we did here was we said, let's assume that the matrix supplies the barrier. And so you've got this, again, cross diffusion, degenerate diffusion, logistic growth, and then the matrix is degraded by the tumor cells. And that would be through things like MMBPs and degrading enzymes. Then the question is, how does this model behave? Now, if M is 0, we have the Fisher equation, Fisher-KPP equation, which we teach undergraduates. And we know there's a unique minimum wave speed. And if you start off with initial conditions compact support, that's the speed that you get. And the speed is 2 root dn times rho. Now, as soon as you put this in, the whole thing becomes incredibly complicated. And what we ended up doing actually was borrowing nice ideas from Allee and Massia, who are looking for same sort of issue, but in a different context, different type of model to this one. And well, first of all, we introduce traveling wave coordinates, xi equals x minus ct, where c is the wave speed, transform to those, and we end up looking at this sort of wave speed where we say, well, behind the wave, we expect n to be 1, be that steady state, matrix to be 0 because it's all been degraded. Now, far ahead of the wave, and this is where the problem arises, far ahead of the wave, n is 0, and that now means in your system, you have a continuum of steady states rather than a single steady state. So in the typical Fisher-KPP equation, you've got 2 steady states and you're looking for a heteroclinical orbit. Here you've got 1 steady state and then a line of steady states. And so the question is, as you come out, you're also in 3 dimensions, as you come out from the unstable steady state, do you actually hit that line? How do you hit it and where do you hit it? And that we spent a lot of time looking at, and in the end ended up using a shooting argument. I'm not going to go into the detail of it. Basically, you start off here, and here's the continuum steady state, and then you've got to go to there, get there. Here, this doesn't work because this gives you negative values. And what we end up finding is that, in fact, when we have this degenerate state at the front, where n is 0 and m is 1, we get existence of traveling waves with unique for any positive wave speed. However, if far ahead of the wave, m is less than 1, then we find here that, sorry, this is wrong here. This is the m equals 1, yeah. If m is less than 1, which going back here means we don't have the singularity, we're back to Fisher. And in fact, the wave speed would be exactly the way you'd get from the Fisher equation. It would be 2 times the square root of 1 minus m times rho. So what this tells us, because this is a problem of, there's a lot of work being done in mathematical biology on very, very important work on what happens, how sensitive is your model to parameter values. I think an equally important, if not more important, is how sensitive are your results to the functions you choose. You can argue that this should be 1 minus n. You can argue that it should be a constant. You can argue that it should be 1 minus m. You can argue that it should be 1 minus m minus n. How do your results depend on those assumptions? And what we show here is, in this case, the results are independent of the assumption as long as far ahead of the front, m is not equal to 1. So apart from that singular case, the model is really not sensitive to the nonlinearity that's in place. And so we've done a number of studies of this sort of form by putting things like 1 minus n in there, extending this a little bit in order to show that. Yeah?
Is the idea that there's a critical matrix density below which things are fine, and then above this critical matrix density, you get things to stop?
Well, no, they're sort of saying that from the point of view of, I mean, you're always going to get a traveling wave, what this analysis says. But so what it's saying from a mathematical point of view, that if you're away from this critical case, then it doesn't really matter how you model this. And so actually what you should do is just get rid of that m. And then it's a simple thing to model. But if far ahead m is 1, then you need to take this into account and keep it there. So it's really from a mathematical convenience point that it's telling you under what conditions are your results robust to your underlying assumptions. So whether you say, oh, cells are just diffusing, or whether you say, oh, there's a barrier, which is the matrix, the net result will be the same unless the matrix is at 1. So from that point of view, there's a structural stability. That's really what we're looking at. So it's more looking at the structural stability of the model rather than the implications on the biology. But the implications on the biology then are that your results are a bit more robust. Okay. Now, another thing is when you look at the models that people put, where they put logistic growth, 1 minus n, and people argue that that's because of space filling. They argue that as n goes to 1, there's no more space left, and therefore the cells can't divide. Okay? Well, if there's no more space left, how can the cells move? So you write down the equation, du by dt equals d2u by dx squared plus u into 1 minus u, and you say when u is 1, there's no space. But what about the d2u by dx squared? It can still diffuse, even though there's no space. Now, you could argue and say, well, actually, the resource limitation is food. And so the 1 minus u is related to food. But what we thought we would do is let's systematically derive the equations based on volume filling. So we say that for a cell to move, there has to be space. For a cell to divide, there has to be space. And space is occupied by cell and matrix. And so then we come up, and this is work with graduate student Rebecca Crossley, with this model. And now you see we get this extra term in here that we didn't have in the previous model that comes from the space filling. And then there's a term there from the space filling.
Yeah.
That one there, that basically what that really boils down to is that the cells, they're pushing the matrix. So as the cells divide, they'll push the matrix. Because they're sort of dividing and trying to move through the matrix. So they'll push it. Because if the matrix has got volume and then the cell divides, or the cell moves, then the cell pushes the matrix. Yeah. But I mean, this comes out from writing down a master equation where you say that, what's the probability that you have n cells at a certain time, and that depends on whether you can divide or you move, et cetera, and probability that you can divide is based on the fact that there's space available, stuff like that. And then when you coarse grain that, take the limit, you end up getting this type of thing here, okay? And so we've been analyzing these types of equations, and in fact, it turns out we have not been able to do a proper traveling wave analysis of this, except in certain limits, like where lambda goes to 0 or lambda goes to infinity. So you get the traveling waves, as you would expect, but the analysis becomes very, very difficult. So what I'm doing here is I'm just sort of trying to point out that if you look at things that people have ignored in these models to try and come up with some general results, that some of them you can come up with, some you can't. So whereas for the previous model, we were able to say, it doesn't really matter whether you've 1 minus m, except if m is 1, and we can analytically show that. For this one, with full volume filling, we haven't been able to show that. So now another thing I want to come up with is cooperation. So tumors are heterogeneous. And the tendency tends to be that, oh, that's just tumors. They're just heterogeneous. And we asked the question, well, could that actually be a strategy for the tumors to survive? So instead of having a supercell that can do everything, maybe you've got some cells that can divide, some cells that can move, some cells that can break down the matrix, et cetera, et cetera. This is an extension of the go or grow model first proposed for glioblastoma many, many years ago, where the idea was phenotypic switching. You move for a while, then you stop, have a cup of tea and divide, and then you move for a while, stop, divide, go or grow. So this was motivated by work, a graduate student, Maxi Strobel, and colleagues at the Moffitt Cancer Center, Magdi, who gave us experimental results, and Bob Gillies and Sandy Anderson. And this was motivated by some of these pictures that Magdi had, which were, this are ductal carcinoma, where the green are cells that are producing lactic acid, and the purple are cells that produce MMPs. So in other words, matrix degrading enzymes. So what we did here was we said, okay, let's assume there are two barriers to the movement of the cells. One are normal cells we call stroma, and one is matrix. And then we'll have a tumor cell population that divides and produces lactic acid, and we'll have a tumor population that degrades the matrix. And the equations are exactly how you would expect, based on what we've done before. Now we're going to sort of ignore the volume filling and look at a simpler version. So basically the idea here, stroma divide, get killed by lactic acid. Lactic acid is produced by the TA cells. The TA cells, I mean, the TM cells degrade the matrix, and that allows things to move. So if you look at this, if there were no TMs, matrix wouldn't degrade, therefore the TAs wouldn't move. If there were no TAs, S would be at 1, and therefore the TMs couldn't divide, because this would be 0. We need to rescale these things, so we'd have that to be 0. Okay? And if you look at this here, this is Lotka-Volterra system. And we all know the Lotka-Volterra system, you can either get competitive exclusion or you get coexistence. And so again, you don't really need to do the numerical simulations to figure out what's going to happen here. In the coexistent regime, you're going to get invasion, because you need both. In the competitive exclusion regime, you're not going to get movement, not going to get invasion. Okay? Here's our traveling waves. We did all the analysis, not going to go through this. Instead, I'm going to just use this cartoon. Mutual coexistence, exactly what we expected. Fantastic invasion. Competitive exclusion of the lactic acid producers. No invasion, that's what we expected. Competitive exclusion of the matrix degraders, we expect no invasion. Wrong. We do get invasion. We get a niche formed here, which is something we didn't expect, and we haven't actually understood this. And then we get mutual exclusion, which again, no invasion. So of all these results, this was one that we didn't expect. But now, if you allow phenotypic switching, the go or grow thing, where you can switch from one type to another, then in your Lotka-Volterra model, once you allow switching, competitive exclusion is no longer a steady state. The only steady state is coexistence. And in this context, you will always invade. So what this model is telling us then is that if you have phenotypic switching, you will always invade. If you don't have phenotypic switching, you'll invade sometimes. And what I mean by always and sometimes, I mean in the parameter space. Phenotypic switching, in all of parameter space, you will invade. No phenotypic switching, then in some of the parameter space you will invade, and in other areas you won't invade. And then, I won't go into this detail, we then asked the question, what would happen if you had a generalist population which can do everything versus specialist populations cooperating? Which is the better strategy? Is one better than the other? And what we found, and we did this, actually in this one then we did go back, put in all the volume filling, etc., etc. I'm not going into the detail. What we ended up finding was with phenotypic switching in, so you always get invasion, there are certain regions of parameter space in which you'll get one, and in which the generalist is the better invader. Other areas of parameter space where specialists are better invaders. And then one of the things we did here was we asked the question, if we have different environmental cues that lead to switching, what types of structure will we get in the traveling wave? And for one of those strategies, what we found was the proliferative cells were at the front and the movers were at the back. And we're just playing around here. You know, we're not thinking of an application here. We're just playing around. If you have different stimulations, what do you get? And ironically, while we were doing this, another group were working in melanoma, and they had been trying to figure out how come in these melanoma cells, the proliferators are at the front and the movers are at the back. Doesn't make sense. And we playing around, just doing a bit of mathematics for the fun of it, actually said, oh, this might be what's happening in the melanoma. Yeah?
Please make sure that switching, this is symmetric switching strategies.
No, yeah, this is switching between cell types. Yeah. So you're yeah, no, it's not symmetric, no. So you could have, well, if it's constant switching, it doesn't have to be the same constant going in another direction. Yeah, it can change. Well, we haven't really explored that in the detail. I mean, don't know where I don't have the results written up here. What we found was it actually depended more on whether your switching depended on the external cues. So for example, if there's more matrix, then you might think you should be better off if you switch to matrix degraders. Yeah, that's right, yeah. Okay, so I'm now going to leave that and go on to talk about something that's more embedded in the biology, and I wanted to dedicate this to a memory of my very good friend and experimental colleague, Paul Colessa, who passed away just over 5 weeks ago, and all of this work was done with him. And this is on neural crest, and so this is work done with Paul and also with Ruth Baker. And so neural crest are basically groups of cells that delaminate, undergo AMT, delaminate from the neural tube, and then go off to where they need to go to do their business. Okay. There are 4 neural crests. They do this movement in different ways, and they do it differently depending on the species. Now, this is important because 1/3 of birth defects arise because something goes wrong with neural crest, and also because things like melanoma are actually related to neural crest. So here's a cartoon from Paul, head, tail, cells coming out along corridors, going to where they need to go. Okay, so here come out from here, and they go to here, and the domain is growing as they go. Now, there's been a lot of work done on various neurocrests by Kerry Landman, Matt Simpson, and Don Newgreen. So these are theoreticians, this is developmental biologists. Hirschsprung's disease. So Hirschsprung's disease is where the gut is growing and the neural crest cells are coming along, and it's a race. The neural crest cells have a certain time to get to the end. If they don't get to the end, then the mechanical properties of the gut are screwed up. And the net result is you get Hirschsprung's disease, which means big gut. Now, the obvious thing to think here would be there's something wrong with the genes that code for movement. The cells aren't going far enough. But the cells are also proliferating. And so we know from Fisher KPP that the diffusion and proliferation contribute equally to the invasion. So it could be you're looking for totally wrong genes. And then I'm not going into the detail of this, but they validated their model against some experimental work. And then there's this other model from Roberto Maior's lab, and it's called contact inhibition of co-attraction. And I call this the mathematician at a party. Okay, so I don't know about you, but when I'm at a party and I'm talking to someone, getting on very well, and then they ask me, what do you do? And I say I'm a mathematician, then they move a bit away. And I like them, so I said, yeah, but mathematics is really important. It's really of interest, and we can do all this, we can do all this. And we end up moving across the room. See, so that is attraction. I like this person. Repulsion, mathematician. Okay. And they showed in the gut of the xenopus that they've done mathematical models based on actually going into the detail of all of these, putting in mechanics of these, having them move and push each other, push and contract, etc. And then you show that you can get this type of behavior, which is, notice, a clump moving along. Okay, and people like Kevin Painter and Jonathan Sherritt, they've looked at this in terms of having a reaction diffusion system with a non-local integral partial differential equation, and showed that you can get this type of pulse-like behavior. Okay, so now back to our neural crest. The ones we're looking at are the ones that form this part, cranial neural crest that form this part. And they don't really divide. So we can't use Fisher equation. So things we're going to do, first of all, is we're going to assume that the curvature doesn't matter. And recently we did some theoretical work that showed that we're justified in doing this. But we're going to show that the depth doesn't, we're going to assume the depth doesn't matter. Depth is about 3 cell layers thick. And we don't have any evidence to support that. It's a simplification. So this is going to be a flat rectangle. And this is the growth of the rectangle from data. Grows from, roughly speaking, 200 microns to 1200 microns over the space of a day. So the prevailing model hypothesis was cells delaminate from the neural tube. If they were to move randomly, a simple calculation shows they'll not reach the end in a day. It'll take them much longer. Secondly, they won't form the stream that's observed. So the prevailing hypothesis was they'd found VEGF being produced by the environment, and the neural crest cells, they love to eat VEGF, so they come out, they consume the VEGF. They're chemotactic, so they move up the gradient of VEGF. So this is a cell-induced movement in response to gradient. So what we did was test this. So we wrote down a PDE for the chemoattractant. So this is sort of non-dimensionalized. And so we've got 1 over L squared here. And that's just a fit to that logistic growth. We've got production. We've got dilution due to the fact that you've got growth. And then we model the cells as discrete entities that have position xi, yi, and consume chemical VEGF in a way that rapidly decreases as you move away from the cell. The form of this doesn't matter, yeah? And no, this is just fitted to the data. Yeah, so this is imposed on the system. And then we've analyzed this model for many, many different parameter values, and the behavior we get is robust, and it doesn't work. domain is growing, the cells come in and they secrete, they end up forming a localized. Oh, one thing I should mention here is that we need boundary conditions. And what we assumed here was that the chemical was fixed to 0 at the boundary, so that gave an internal gradient, so you couldn't leave. Later on, we assumed 0 flux boundary conditions, so you couldn't leave at all. And there's also a robust to that, but I'll come back to boundary conditions a bit later on. But basically what happens is you get a sort of clump of cells moving. Now, you could argue, well, if you did enough simulations, this is stochastic and everything, maybe this wouldn't happen. But if you look at the deterministic analogy of this, which is a Keller-Siegel model, which was N moving up chemical gradient, this was proposed particularly to describe clumps of bacteria moving. And this model can only give you the sort of behavior that we want in the singular case where sensitivity to the chemoattractant goes to infinity as the chemoattractant goes to 0. A fun mathematical thing, but probably not very realistic biologically, where you're most sensitive to signal when there's signal isn't there. So Louise Dyson, who is the student working on this, she said, well, okay, the cells at the front, they can assess, they see VEGF, and so they move in response to that. What do the cells at the back see? They just see the cells at the front. So maybe they just grab onto the cell at the front and move along. And then if you do that, you get these movements. So then we went back to Paul and we said, you know, these cells that you told us about, are they all the same? And he said, I don't know. Good question. I'll do some analysis. And this is going back before we could do single cell RNA-seq. So he took quartiles, did single cell, did sequencing of these groups of cells, looked at a whole pile of genes. What he found, the cells at the front upregulated MMPs, which break down the matrix, upregulated receptors to VEGF, so they could move up the gradient, and the cells at the back upregulated cadherins that made them more sticky and more adhesive. What our model predicted. But it's more complicated than that. There's a gradient of adhesivity. And that's something we're looking at. Then we asked the question, okay, you could have the cells stick to the front. You could also have, in fact, what he also found was that the leader cells upregulated aquaporins that drilled a hole through the environment. And so we could have a tunneling that then the cells at the back, if they get into this tunnel, they can move. There's also mechanical cues that are possible. And so we did a pile of experimental validations here. There's a number of ones where the model and the experimental model and the theoretical model agree. I'll just show one example. What happens if you remove the followers? Here's the experiment. This is under normal conditions. This purple is when you remove the followers. And what you find is the leaders are spread out more. And this is what you get in the theoretical model as well. And that's because there isn't any volume pressure. Now, here's one where I think we learn more is when the experimental model doesn't agree with the theoretical model. What happens if you take followers from a host and stick them, from a donor and stick them way ahead front of the host? Our model says followers, they're the ones that follow. There's nobody for them to follow now, so they just move randomly. Totally wrong. They become leaders and actively move. So this means we propose they've switched phenotype, and they switched phenotype in response to the environment. Next experiment, obvious, put beads of VEGF along the corridor and see what will happen. Well, you don't need to do a mathematical model to do that. You just need, I would say, maybe 2 brain cells to figure that out. The cells that are here, they're going to keep following the gradient. The cells, the followers, are going to suddenly go, oh, VEGF, I love VEGF, eat VEGF, create a gradient and move there, and you go and get a split of the 2. And that's exactly what happens. Okay. One thing I pointed out was, or one thing I actually tried to hope you didn't see, is in our model, some of these cells, leaders get too far ahead and leave the followers behind. So then we asked Paul, is there something that controls the leaders so they don't move so fast? And what he found was something called Dan. Dan is expressed right next to the entry into this corridor, and Dan slows down cells. So how's that going to work? The leaders slow down, the followers also slow down. You're still going to have the problem. If the leader is too fast, it slows down a bit, follower slows down a bit, still not going to work. Except by magic, after the leaders go through the Dan is no longer expressed. So the leaders slow down, and the Dan goes away, and the followers don't slow down. This is what I love as a theoretician working in biology. Biology is just magic. It's fantastic. And then, of course, the obvious experiment, confront melanoma cells with Dan, and you slow them down. Other signaling cues, so since, go through this very quickly. Another thing that Paul noticed, this is work that Duncan Martinson did, was there are these puncta fibronectin, which are balls of fibronectin that are present. And as the cells move over the fibronectin, they align it. Now, the fibronectin has an adhesive gradient. So haplotaxis, you move up the adhesive gradient. Also, the alignment offers a directional cue. So what Duncan did, here we just want to focus on that. So we don't have domain growth. We don't have chemotaxis. These dots are the puncta. These black cells are the leaders. As they move over, they align the puncta. And this is a mechanical model where then the followers, there's mechanics going on. I'm not going into the detail, where they're pulling on forces, there's volume exclusion, everything's going on, and then they pick, there's a radius around which the forces affect their movement, and then they move. And what you see happening is you get a much more coherent movement than the one we got when we just had stickiness. So coherent that even if the leaders go off in the wrong direction, they take a big pile of people with them. We don't have any zero flux boundary conditions here. Okay, so Duncan did a huge amount of parameter sensitivity analysis in all of this. I won't go into all the details, but what he concluded was contact guidance and matrix remodeling, they are sufficient to enable the collective migration. So as long as you give the cells at the front the correct polarity, these together are enough to give you the movement. So the very last thing I want to discuss, and this will just take a couple of minutes, I'm sorry for running over time. Remember the boundary conditions. How are the cells confined to the corridor? Well, we just didn't allow them to leave, so we cheated. So here's what we did: barrier, barrier, and you get nice movement. If you didn't have barriers, they go all over the place. If you had an internal barrier of a certain length, then it's sort of okay. So the question we then asked was, how big does this barrier have to be in order for this, the minimum size, in order for this to work? And Raza, who was a student who worked on this, came to the conclusion 500 microns. So then we asked Paul, if you look within 500 microns of the neural tube, do you see anything? And the answer was yes. Collect 12 and trail. These are two proteins that are expressed within 500 microns of the neural tube. And I won't go into the detail of what these do. Happy to talk about it afterwards. But if you put in what these do, namely, one of them gives rise to amoeboid motion, which means you move more slowly and you move together. The other one causes the filopodia to go absolutely mad, to be so many filopodia, and they're so much longer. And we propose that that means that if you get off track, you've got a better chance of finding your way back, because you've got longer arms to sense where you should be going. And they had noticed that sometimes cells go off and then they come back, and they hadn't known why. This, we think, is why. And using our model and putting all that into the model, we end up getting a synergy. So each of those on their own works quite well. Put them together, they work really well. And this is work actually that we submitted a few weeks before Paul passed. And very ironically, we got the acceptance of this on actually the day of Paul's funeral. So that was a very sad occasion. And Sam is the student who's worked on this. So the actin synergy, now, sometimes it doesn't work. This is stochastic. And so sometimes they'll go like this, and then they'll just go off. Which is a good thing, because that actually happens in real life. So the main criticism we got when we had the model with zero flux boundary conditions is that your model doesn't allow this to go wrong sometimes. Now, when we put this all in, we get it going wrong sometimes. Okay, now I'll just point out some other things, and then I'll stop. The growth is not spatially uniform, and this relates to some of the conversations I've had earlier on today. The growth is spatially non-uniform, and it is stimulated by the neural crest cells. So I know what you were getting at, Vijay, when you said, have you imposed L? Yes, we cheated. It really depends on the system. And that's something we need to look at. It's something that you guys are looking at as well. So maybe there might be some synergy there. There's a gradient of phenotypes, and we really need to model the mechanics properly, right? And we're not doing that either. And there again is something where I see there might be synergies going on. And note there's so many similarities between this and cancer and angiogenesis. In cancer, there are leaders and followers. There's phenotypic switching, for example, in angiogenesis, tip and stalk cells switch. We know a bit more about that through Notch delta signaling. VEGF plays a huge role in cancer. So huge number of similarities. So I'll end there by acknowledging the Royal Society Yusuf Hamid Visiting Fellowship, which was actually sponsored by Mohit. He's the one that found it, and then we applied, and then he's looked after me. And thank you again for your attention, and sorry for running over time. Yeah.
People online, any questions, comments? I wonder if the boundary conditions matter for the traveling wave part. I mean, I don't know what you did in the direction in which the waves are traveling. What boundary conditions do you use?
So sorry, in
In the first two parts when you showed the traveling wave?
oh yeah, in the first two parts we just have because we just have it in one dimension, we have just the boundary condition is like flat at either end, so zero flux because we model infinite domain, and then we just have that you start off at 1, 0, and you go to 0, 0 basically, or 0, 1, depending on the matrix. And then for the two-dimensional ones, we have zero flux boundary conditions in that. Yeah. In fact, one thing we've been trying to do on this one is to look at a non-local model for the neural crest. But then there, as soon as you have a non-local model, because we do have this boundary, that becomes a real issue. I mean, how do you, with a non-local model, deal with the boundary? We haven't figured out a nice way of doing that at the moment. In another context, we've used non-local models, but we've just had periodic boundary conditions. But here, given that, I mean, if you wanted to model the contact guidance, et cetera, or the mechanical effects by a non-local system, then you really do need to worry how you're going to deal with the boundary conditions.
Questions, comments? Okay. All right. If not, let's thank Philip for a very nice talk.
Okay, thank you.